Hello, my name is April. I helped create this module, and in this video we will walk through how to use a genome browser to view different genomic data and to learn more about protogenes. I'll be walking through the module and answering the associated questions using the protogene ORF 14869. To find the modules and the associated worksheet, go to the Orphan Gene Project website, and then to the Lab Modules and Adopt a Protogene Project tab. Open the guide and worksheet, and on the worksheet you can go ahead and make a copy so that you're able to type on the document. Okay, so then going back to the module as a guide, we can scroll down to the activity. And first we want to find out where our protogene is located in the genome and like what other ORFs it's nearby and so on. To do this, we're going to go first to the ORF information website. So this first link here, and type the ORF ID in the box. In the coordinates table here at the bottom, it tells you about the location of your protogene as well as the strand. So that's the direction of the protogene. And using this information, you can now answer question 1 in your worksheet. So for my example protogene, it's located on chromosome 2. And it begins at coordinate. To visualize where your protogene is relative to other genes, click on the genome browser link that's below the table. This takes us to the JBrowse website. At the top, it gives us the location information, so on chromosome 2 and between these nucleotide coordinates. The plus and minus buttons up here allow you to zoom in or zoom out. You can also drag the screen to the left and right to view other locations. Each row in a genome browser is referred to as different tracks, so they contain different data sets or different information. This top track here is labeled all annotated sequence features, and it shows all annotated genes and other sequence elements as these rectangles. If you click on one of these genes in this annotated sequence feature track, a window will pop up that gives you more information about the gene. So in the description, it gives you information about its function, it gives you information about the length, its sequence, and other annotations. In the track below is this track called Watchholder 2023 Translated ORFs. And in this track, it contains all translated sequence elements. So it contains both annotated genes that are found in the all annotated sequence features tracks, as well as containing protogenes, so these other open reading frames that are not annotated as genes. So in the center of your screen should be a rectangle with your ORF ID below it. Like before, if you click on the rectangle in this track, it will open a new window and tell you more information about the ORF, such as its length, its sequence, and its evolutionary status, so whether the ORF is conserved or found in other species, or if it's evolutionary transient or evolutionary novel. And this other attribute called score tells you the confidence level that the ORF is translated. Okay, so now from this, we can go ahead and answer questions 2 through 6 on the worksheet. Okay, so question 2, what is the length of your protogene in base pairs? So how many amino acids is this? So this ORF is 150 base pairs long. Each amino acid is made up of 3 nucleotides. Therefore, this ORF would be 50 amino acids long. Okay, question 3 is your protogene on the plus or minus strand. And so from question 1, we know it's on the minus strand. And if we look at it in the genome browser, we see that the arrow is pointing to the left, meaning that it is it's going from right to left. Therefore, it's on the minus strand. Okay, question 4 is what is the nucleotide sequence of the second codon for your protogene? Okay, so we'll need to look at the sequence for this. If we go back to JBrowse, and again we click on the ORF, it shows the sequence here. Each codon is 3 nucleotides, so each codon encodes for an amino acid, and each one is 3 nucleotides long. The sequence of the second codon would be TCC. Okay, question 5. What is the name of the annotated gene closest to the 3' end of your protogene? And what is this function? And is this gene on the same strand as your protogene? So here is my example protogene, and I want to know what annotated gene is like the closest neighbor at the 3' end. So the start of the ORF is the 5' end, And since this ORF is on the minus strand, the 5' end is to the right, so this area here. And the 3' end is the end of the ORF, so this is down here. And if I scroll over and look at this top track for the all annotated sequence features, I see that the closest annotated gene is this really long red one here called PGI1. Okay, so now we need to look and see what is PGI1's function. If we click on the gene, it tells us that it is a glycolytic enzyme and is required for cell cycle progression and completion of gluconeogenic events. Looking at its direction, we see that it's also on the minus strand and it's going from right to left. Okay, question 6 is similar. It's what is the name of the annotated gene closest to the 5' end of your protogene, and what is its function, and is this gene on the same strand as your protogene? And I now want to find the closest annotated gene at the 5' end. So I'm looking for a red box in this top track that is the next closest. So if I scroll over, I see that the next closest one is YBR197C. And if I click on it in the description, it says that it's a protein of unknown function, so there's not much known about this gene. However, that it localizes primarily to the nucleus whenever there is DNA replication stress. And by looking at the direction of the arrow, we can see that it is also located on the minus strand. So it's on the same strand as my example protogene. What's really great about genome browsers is that you can integrate lots of different data sets to look at a specific region. Okay, so now we're going to investigate the translation of your protogene. And for this, we're going to be using these bottom two tracks. So either the watchholder riboseq plus or the watchholder riboseq minus, depending on which strand your protogene is on. So in my case, since my protogene is on the minus strand, I'll be focusing on this bottom track that says minus. And so in this track, it contains the riboseq reads at each position. And for example, I don't see anything over the region where my protogene is, and this is because there are so many riboseq reads covering this downstream gene, so PGI1, that the scale is very high and I no longer see anything. So to fix that, we can either zoom in until we get away from this really highly translated region, as well as we can change the scale to be in log scale. Click this down arrow and select log scale. So if I zoom in on my ORF, I now start to see these different bars over it, and the height of the bar corresponds to how many riboseq reads were found for this position. And the more reads that you find, the higher confidence or the more evidence that this ORF is translated. So when I look over this ORF, there's not too many reads. This is in general not a really highly translated ORF, and from this data alone, we wouldn't be that confident that this ORF is translated compared to some of the other ones. So this is just some reads over the ORF, but if we wanted to have an actual number to gauge our confidence, we can click on the ORF up on the translated ORF track. And if we go to the score section, the score represents like how confident we are that this ORF is actually translated and not just due to chance. The lower the number, so the closer to 0, the more confident we are in it. And the higher the value, so closer to 1 or like greater than 0.5, means we are not confident that this ORF is translated. So here we see a value of 0.02, which is pretty good. However, we're very confident in scores that are below 0.01. Okay, and from this, we can now go ahead and answer questions 7 through 9 in the worksheet. So for number 7, does it appear that riboseq reads are spread evenly across your protogene, or are there spatial patterns and trends you observe? So for example, does there tend to be more reads early on in the sequence or towards the end? For my protogene, there doesn't really seem to be any trend. There's really not that many riboseq reads over the ORF, like in general. So I don't observe any pattern. They tend to be spread out evenly over the ORF. Okay, question 8. In general, do annotated genes tend to be more or less translated than protogenes? Here was my example protogene, and it was not very highly translated. There were very few riboseq reads over the region. However, if we scroll down and look at my protogene's neighboring annotated gene, so PGI1, we see that it has high evidence of translation. So there are lots of riboseq reads, and the bars go up to really high numbers. So for example, like this bar here is 29,000 riboseq reads, and there tends to be pretty even coverage of riboseq reads over the gene. And we click on the annotated gene to get its translation confidence score. We see that it has a score of 0, so we're very confident that this gene PGI1 is translated. So if we go ahead and zoom out, like keep zooming out so we can get a bigger genome-wide picture and try and look at the density of riboseq reads. So here in these bottom two tracks, relative to where these annotated genes are, so these red rectangles, we see many more reads over annotated genes compared to these protogenes. And we can go ahead and answer question 9. So what is the translation confidence score for your protogene? I see that its confidence is 0.02. Next, we're going to look at RNA expression for our protogene. We are going to pull up a different data set in these tracks. So go in the top left corner, click the select tracks button. And in the search bar, we're going to type in Pellicano and select these first 2 boxes. So this is a data set where they looked at the full-length mRNA transcripts in different environmental conditions. So they performed the experiment in both galactose, which is one type of sugar media that can be used to grow yeast, and they also looked at yeast growing in YPD, which is another growth condition that you can grow yeast in. And so they did this experiment because under different environmental conditions, organisms will express or activate different genes. And so we can use this track to see if in either of these conditions that our protogene is expressed and if the transcripts look different. So one thing to note is that before this dataset was put up onto this genome browser, they pre-filtered to include only transcripts that contain annotated genes. And so if you do not see a transcript covering your protogene, that doesn't mean that it's not expressed. It just means it was either not found in this experiment or that it was filtered out. Another thing to note is that you need to pay attention to the direction of the transcripts and make sure that the direction of the transcript is in the same direction as your protogene. So for example, for my protogene here, there are 2 RNA transcripts covering the protogene and they're in the same direction as my protogene, so also on the minus strand. And I noticed that there's a difference between the transcripts that are made when yeast is grown in galactose versus in YPD. We can go ahead and answer question 10 on the worksheet. So do you find transcripts covering your protogene when yeast is grown in either YPD or galactose? And do you notice any difference between the 2 conditions? So I find there is mRNA transcript covering the ORF in both YPD and galactose, and that the mRNA transcript, the one that's grown in galactose, is much shorter than the transcript when yeast is growing in YPD. And lastly, we're going to look at the nucleotide conservation over your protogene. We're going to go back up to the select tracks, and in the search box, we're going to search UCSC. Select this box that comes up, and it pulls up this bottom track here, the UCSC conservation track with these purple lines. Here, scientists use a method called FastCons, and how this method works is that it applies a score to each nucleotide based on how similar that nucleotide is in other closely related species and how similar the nucleotides around it are in these other species. And in this case, a score of 1 means that the nucleotide is highly conserved, and a score of 0 means it's not conserved. And here we see the distribution of nucleotide conservation over the ORF. And if we zoom in, we can see that there are regions that are more conserved than others. The higher the peak is, meaning the more conserved that region is. And then when it's low, it means that region is not conserved or maintained in other species. And when looking at my protogene here, the beginning is somewhat conserved. It becomes much more conserved when we get towards the middle. So we have conservation scores of like 0.8, and then it drops off in this like middle to end region where there's really no conservation at all. The scores are all close to 0. And then again at the very end, there starts to be a highly conserved region with scores around 0.8. And if we zoom out, we can look at the conservation of like the surrounding areas, and we see for the neighboring annotated gene downstream PGA1, it has very high conservation across almost the entire gene. And we also see for these other regions that have lots of protogenes that there's variable amounts of conservation, but it tends to be pretty low. And so now we can go ahead and answer questions 11, 12 in the worksheet. So question 11 is, are there certain regions of your protogene that are more conserved than others? Yeah, so I find that the nucleotides in the beginning of the ORF are lowly conserved, and then towards the middle, there are a few nucleotides that are much more conserved, and then there's a while where there's nucleotides are not conserved at all, and as you get to the very end of the ORF, the nucleotides are again well conserved. And the last question, question 12, how does the nucleotide conservation of your protogene compare relative to the closest 3' and 5' annotated genes? The conservation of the nucleotides for my protogene ORF 14869 are on average much less conserved than its 3' annotated neighbor PGI1, because pretty much all of the nucleotides in PGI1 are perfectly conserved, so they have a score of 1, except for a very small region of a few nucleotides towards the end that have low conservation scores. I can see that PTI1 is a very well conserved gene. My protogene ORF 14869 is actually more conserved on average than its 5' annotated neighboring gene, YBR197C. It has portions of conserved nucleotides, but it has long stretches of not conserved nucleotides at the beginning of the ORF, and a small region towards the end of the ORF that is not well conserved.